Okay, so it's time to move on from our discussions of DNA and chromosomes, the stuff of inheritance, and to start talking about some of the processes by which that stuff, those molecules, get moved around. And there's two processes we need to consider. The first is mitosis, and that's what we'll do today. And then the second one, which is more complicated, is meiosis. We'll look at that in a separate lesson. To understand mitosis and to see where it fits, we need to understand the life of a cell. Notice here we're talking about eukaryotic cells. Prokaryotic cells are simpler, and so they don't need as detailed a treatment. But you can sort of view the processes they use as simpler versions of what you're going to see here. Okay, so we're talking about eukaryotes, and eukaryotic cells have three stages: interphase, mitosis, and cytokinesis. Interphase takes up the majority of the life of a cell. It is doing its job. If it is a liver cell, it is breaking down toxins. If it is a nerve cell, it is sending signals. If it is an eye cell in the retina, it is picking up light. And of course, they're living while they're doing that and respiring. And they're also growing in size from their junior infant size to their full mature size. Mitosis, which is what we're interested in today, is when the genetic material gets copied and prepared for separation into separate cells. And finally, cytokinesis is when the parent cell actually splits into the two new ones. Now, cells are fantastically complicated things, but once we assume that a cell is doing its job, we can observe it under a microscope, doing whatever it's doing, and most of the time nothing much seems to change. When we get to mitosis, however, there is a dramatic change, and it is a complicated change, and that is why it gets a special name and why we have to take some time to discuss it in detail. Interphase and cytokinesis are not technically part of mitosis. The cell cycle is the life cycle, and we divide it into three stages because they're distinctive. However, even though that's true, I'm going to discuss them all together under this PowerPoint of mitosis. Okay, so although a cell cycle has three stages, mitosis itself has 5 phases. We've already seen interphase, and I've already told you it's not technically part of mitosis, but we do need to treat it with the other phases. Then we have prophase, we have metaphase, anaphase, and telophase. And you can see that if you need a little memory device to remember the phases, IPMAT, IPMAT, is a little acronym that you can use. So let's actually have a look here at the different phases in detail. We start with interphase, and as we've already said, cells at this time are doing their normal jobs and growing. Remember that the purpose of mitosis is to take DNA, copy it, and divide it perfectly in preparation for the separation of two halves of the cell into two new smaller cells. So what's the DNA like at this time? Well, it's not, during interphase, DNA is actually not in chromosome form. It's in long string form. And if you were to look at it under a microscope, you wouldn't see the strings because they're far too narrow. You would just see a dense mass, and that dense mass is called chromatin. The replication, which we have talked about in an earlier lesson, occurs at the end of interphase in preparation for mitosis. Now, throughout this lesson, I'm going to show you two kinds of pictures or images. The first is a diagram, a simplified version of what's happening in mitosis, and then I'll give you a photo, a light microscope photograph of a plant cell actually going through mitosis. And up the top here, we can see there's a nucleus in the middle. We can see a nuclear membrane around the outside of the nucleus. There's the plasma membrane, the cell membrane around the outside there. Down here, these are the boxy cell walls of the plant. There's the nucleus in the middle, still with its membrane. And you can't see any squiggly X shapes or wormy shapes or sausage shapes because there are no chromosomes. You've only got chromatin. Well, let's move on to prophase, which is the most complicated phase of mitosis. This is the phase in which there are the most things that you have to remember. And the first thing you need to remember is the nuclear membrane, that's the membrane around holding the nucleus together, at this point begins to disintegrate. So all those phospholipids let go of each other, fall apart, and there is no separation between the contents of the nucleus with the DNA and the rest of the cytoplasm. During prophase, the chromatin, that's the long strings of DNA, get knitted into chromosome X shapes. Another really important thing that happens during prophase is that a new structure, which was not there before, called a spindle, forms. And a spindle is not a single stick or a single tube. It's actually a large bundle of fibers made of proteins. And we're going to see what they do in a minute because they're extremely important. The spindle fibers attach to the centromeres. Remember, the centromeres are the protein staples that hold the two chromatids together. Now let's start with the spindle. So in this image, you can see blue squiggles and a sort of red fuzz that looks a little bit like an onion, a red onion cut in half. That red onion stuff is actually a large bundle of protein fibers. Note that this cell does not normally look like this. It's been dyed with a number of different stains to give it, to make it very clear. But that's a light microscope image of a cell in prophase. And notice all the squiggly, wormy, sausage-shaped things in there. They don't have an X shape because they're all lying on top of each other. That's the chromosomes in actual chromosome form. If we go back to the version that I had before, here's our simplified diagram, the plasma membrane around the outside, and here we have the chromosomes have formed in the middle and the nuclear membrane hasn't quite broken up yet. Here's a real cell. We can see the boxy cell wall. We can't really see a nuclear membrane, but we can see the chromosomes, and they are still fairly tightly bundled into a circle in the center of the cell. So again, this will probably be early prophase. Well, the next stage is metaphase, and metaphase is much simpler to understand. There is no nuclear membrane. In fact, the only thing that happens here, the distinctive thing, is that the chromosomes get lined up at the center of the cell, and the center of the cell is called the equator. How do they get there? Well, those spindle fibers, those protein fibers which are attached to the centromeres, now push and pull the chromosomes until they arrive and line up along the equator. If you want a little memory device for metaphase, something to distinguish it from all the other phases you have to remember, I always think of M for middle and for metaphase. Here's our diagram. Notice here we have our cell membrane around the outside, but there is no nucleus anymore, no nuclear membrane at all. We have instead our chromosomes lined up down the middle, and we have these little lines representing the spindle fibers, and now we have that same cell before, but it's now in metaphase. You can just see the cell walls on the outside, and there are the chromosomes lined up in the middle. This is an excellent photograph. It's not a painting, it's a photograph taken under fluorescence microscopy. So again, like in those other pictures, they dye the cell with, in this case, fluorescent dyes, and then they shine a ultraviolet light on it, and using very high quality photographic gear, a number of researchers were able to get this picture. Again, we can see the red spindle fibers, and we can see the chromosomes in blue, and this picture won an award for science photography. All right, let's move on to anaphase. So now that the chromosomes are lined up in the middle, the job of the cell is to make sure that the components of those chromosomes are actually dragged to either end of the cell. And if you recall from the last lesson, each chromosome has two arms or two legs or two sides, whichever way you like to look at it, held together at a protein centromere. And in this case, these chromatids now get separated. And again, the separation happens because of the action of the spindle fibers. So again, we can see our diagram. There is no nucleus in the middle, but we are seeing the chromatids being dragged to either end. And now on the bottom here, here's our plant cell. Notice that in the diagram at the top, the poles of the cell, or the ends of the cell, if you like, one there and one there, are east and west. Down here, the poles of the cell are north and south. So in this case, the cells, the chromatids, sorry, are being dragged upwards and downwards as opposed to left and right. And finally, we get to telophase. This is really the end of the process as far as the separation of DNA. Now the chromatids have been dragged to the end of each of the cell and are at separate poles. The DNA starts to disintegrate from its chromosome form back to its chromatin form, and new nuclear membranes start to form around the outside of that genetic material. Again, we can see our plasma membrane around the outside. We can see two nuclei, but we're beginning to lose some definition on our chromosomes. And again, here, there's our original cell wall. We've now got two nuclei. We're beginning to lose definition on our chromosomes. And in both cases, you're beginning to see some kind of division between the two cell halves. Now that division is actually cytokinesis. That's all cytokinesis is, the actual splitting of the cell. But because of mitosis, because of the careful separating of genetic material, this cell and this cell are now going to have a perfect copy of the information and approximately half of the cytoplasm. In the case of animal cells, there will be a protein or a series of proteins which pinch off a little bubble in the middle, or sorry, pinch the cell off in the middle, creating two separate bubbles. And in the case of a plant, it grows a new cell wall across the center. And now cell division is complete, and our two separate cells will now start to grow, and they will perform whatever job they are supposed to perform, just like we talked about earlier on. And of course, that means they are now in interphase. And so the cycle is complete, and after X number of months or years, the cell will divide again and go through the whole process once more. Now, I've put together all of the diagrams and light microscope pictures that we have been looking at throughout the description so far, so you can see them next to each other. So we have interphase, nuclear membrane in the middle, but no chromosomes, prophase, nuclear membrane, but we can see the chromosomes, metaphase lined up in the middle, M for middle for metaphase, anaphase, the chromatids get dragged to either end, and then in telophase, nuclear membrane starts to form, or two nuclear membranes start to form. The DNA, the chromosomes fall back into their chromatin form, and then finally cytokinesis splits the two cells two sister cells, one from the other. And there are the pictures. Again, you can see the light microscope images down the bottom there. I strongly recommend that you click on these links and have a look at the two examples of mitosis. These are not animations. Sorry, I beg your pardon. The first one is an animation and the second one is a time-lapse photography of an actual cell undergoing mitosis. And I think they will make my static descriptions much more clear when you see the actual process happening in front of you. I'm going to finish with a set of examples because I think it's very helpful to get familiar with the shapes of each of the phases of mitosis, each of those 5 stages, IPMAT. Because what's going to happen is when you see an image in another textbook or in another context, it's not going to look like the examples I've given you. After a while, you get familiar with the shape and you can very quickly say, oh, that's metaphase because they're lined up in the middle. Oh, that's anaphase. Oh, that must be prophase. But in the beginning, it's hard to do that. So I'm going to give you some examples. This first image is in fact a set of 10, which are hand-drawn by a late 19th century, early 20th century biologist. And I'm going to go through them briefly. Here we have a nucleus in interphase, early prophase, late prophase. You can see the chromosomes very clearly. Here we can see the nuclear membrane has broken down, still with our prophase chromosomes. E, they're starting to move. A little bit confusing, I have to say, E. F, we see a very clear metaphase. G, we're beginning to see anaphase. There's another example of anaphase in J as the chromatids get separated, and an even better example of anaphase as the chromatids reach their separate poles. And finally, we have telophase here, and we can see a nuclear membrane beginning to form, and though the DNA is still in chromosome form. Here's another example of fluorescence microscopy. I'm not going to go through them in detail, but they are in order. So interphase, prophase, and all the way through to telophase there. And finally, I'm not going to dwell on this either, but if you want to get practice at looking at mitosis, then there's 5 separate examples there for you to review, each of them being labeled. Some of the images are missing, but you'll get the picture. All right, once you've done all that, you can do a couple of questions in Pearson, and if you need to review any material, get a new angle on this stuff, you can get that from these pages in Chapter 6 of Pearson.